Good morning, everyone. I hope everybody had a chance to grab a cup of coffee at the networking break before the session. Let us begin. First of all, I'd like to welcome you to today's session, all about the challenges of long-read sequencing, sample prep, and analysis. We have a vendor talk from NABSIS that we'll start with, and then we'll have a brief lightning talk from Joan Finn. And then we have 3 great presentations, and hopefully we can follow up with some discussion and, some Q&A. Just a few housekeeping rules. Please silence your cell phones. And photography is permitted. Recordings will be available after the session. Social media policy is that everything can be shared, and if you decide to share, use the hashtag ABRF2024. We ask that all questions for the sessions remain for the latter part of the session after all of the presentations are done. And just to wrap up, make sure that you rate both the session and the lightning talk on the app 1 out of 5 stars. All right, come on up.
Okay, thank you, Kelly. And I'm not going to actually be talking about long-read sequencing, but long-read genome mapping. So it's a little bit different, and it is applied mainly it can be applied for assemblies as well as for structural variants, which is what I'll be talking about today. And if you're interested in long reads, chances are you're interested in structural variants as well. And there'll be an expanded version of this tomorrow at 8 a.m. for breakfast. So if you want a free breakfast tomorrow, you can see these same slides as well as a number of others, also booth 311. So NABSIS does electronic genome mapping. There are other methods, is optical genome mapping, and we have adopted electronic because it's higher resolution, lower cost. It's complementary to short and long read sequencing, so all the talks you'll be hearing later on can be applied, that kind of technology can be applied here as well. We have a strategic partnership with Hitachi and several instruments out with the early access user base. And again, the main function is to look at structural variations. And as you know, there is no single best method for looking at structural variant detection. Here are 2 papers just in the last month or 2 that compare all, not all, but certainly many of the different analysis systems for looking at structural variants using the short reads or long reads. And the conclusion is it depends on the nature of the structural variant. Is it insertion, deletion, translocation? How big is it? Where is it? So there is no single method to really get at this. So having an orthogonal method like electronic genome mapping is highly beneficial because it really approaches the whole problem from a different perspective. And next-gen sequencing, again, as people here well know, you have the short-read sequencing and long reads, which typically are 10 to 50 kb, whereas the ultra-long DNA we use, that is used in optical mapping, tends to be 50 to 500 kb, and you use fluorescent labels to identify particular sites within the genome. With electronic genome mapping, it's used sites similar in principle to Oxford Nanopore, where you're looking at current blockade. We put a piece of DNA into a nanochannel, and that DNA blocks the current, and so you can measure that DNA blockage. And then with specific tags, you can again look at additional current blockade, so you can identify where the tags are, and then assemble a de novo in the absence of a reference, where you can do remapping to a known reference should you choose. And just going to show you essentially what it looks like in real time. So we have, this is the original detector, which had just a single channel with 2 inlet and 2 outlet ports. The DNA is driven through the detector region with a pressure and a voltage gradient, and so the DNA will go through there. And I had said that we do electronic genome mapping, and almost always that is true, and this is the one case we use optical mapping, where we have labeled the DNA. So this DNA is fluorescently labeled, and this is what it looks like when the DNA is going through the nanochannel. You can see especially the long pieces start off going in slowly, and then as the longer DNA goes in, it will speed up, and so that is taken care of in the analysis. So this is a DNA about 400 kb long. You can see it going through, and it speeds up as it goes through. And you can think of it as being on a roller coaster, that when you start in the front of the car, you're going slow down that hill, and as the rest of the car gets pulled through, you speed up and head through. And it's the same principle here, and so we account for that in the analysis system. So right now we have 256 nanochannels in parallel. It's the same sample going through all 256 channels. And the reason for having that many channels is so we can do a whole human genome in a single run. One of the things that will be improved over time is using more channels so you can get even higher coverage over time. But right now, 256 channels is sufficient for doing a whole human genome. And so again, later today, or later this session, you'll hear more about the DNA extraction. We use the NEB Monarch kit for high molecular weight DNA. It's very easy, robust, and generates very good quality DNA that can be used here. The sample prep for running it in the instrument is less than a day, and then you simply load it with a pipetter, and then you run it overnight and detect the tagged molecules. What you get out at the end of the run is a set of times for the tags, and then you convert those times into distances. And because those distances can be highly variable, and there's an average distance of about 4,000 bases between each tag, but some can be as few as 100, 200 bases, whereas others can be 10 or 20,000 bases. Having a pattern of 6 or 7 different tags makes it unique on the human genome, unless it's something that's highly repetitive, which obviously sometimes happens. But you can, again, with a fairly short fragment, know exactly where it came from based on the pattern of tagging. And then you can either do variant verification, where you have a known variant that you'd like to look at. Now, is it there or not? You need less coverage for that. And then if you want to do structural variant discovery, if the whole genome, it takes a little bit more coverage, and you can do a reference-free assembly. We are focusing on human DNA at the moment, but there's no reason why you couldn't use it for other species as well. And again, this is the actual instrument is one in the exhibit hall if you'd like to see it. Just load it with a pipetter, and the detectors are disposable. We use one per sample. And again, this is what it looks like. It's fairly small, and that data is all measured. This is what the data actually looks like when you get it. So these are 2 different molecules. The top one is a chunk of centromeric DNA that's greater than 200 kb. And you can see the tags right every 1,800 bases here, and so you can place that on a particular centromere. And then the bottom one is actually a back that covers the human FSHD region, and the let me just show here. You have repeats that are 3,300 bases. Because we have one tag in each of these repeats, you can just count the number of repeats. And again, the distances here are all the same, and you can actually see that in the front part of the molecule, you have a wider spacing and wider tags. Toward the other end, there are narrower tags. And this is actually an advantage. It helps you with detecting false positives, because you can easily distinguish adjacent or overlapping molecules. By the tag width, you know where the molecule should be. And you also have no implication, no ligation. You're looking at double-stranded DNA, so you're not affected by secondary structure that can be a problem with other long-read technologies that can impact the ability of DNA to go through nanopores. And we can look at all the different types of variants. I won't go through that right now. And again, there's the different parts of the instrument. And again, if you'd like to see this, we have it. The reagents go up here, the detector here, and the waste comes out there. And again, there are 2 pipelines for discovery, either verification of known samples, which is simpler, or you can do the whole genome discovery. And this is in collaboration with Hitachi, who have generated software called Human Chromosome Explorer, and that assembles the DNA and matches it to the human genome. And it can be used for all kinds of different applications: functional genomics, rare disease, cancer research, cell and gene therapy for looking at insertions either on target or off target, look at highly repetitive regions like the CYP genes, and also we're completely positive that once this is out there and used by a lot of people, there'll be lots of other different applications. And I'd like to thank you. This is DNA going through our nanochannel right here, at least an AA version, and not an AA, an AI version of that. Yeah, if AA, it would be a little bit different, I guess. But again, it's all based on current blockade and electronic measurements so that you can get very precise measurements. You can get a resolution down to about 300 to 500 bases with this, whereas with optical mapping, you're talking 3,000 to 5,000 bases, so that having that higher resolution allows you to do things like repeat expansions and other things that would be challenging to do on other systems. And that's as fast as I can do this talk. But again, if you'd like to hear a much more detailed one, you can have a free breakfast tomorrow morning in Minnehaha, and or visit our booth at number 311. Thank you.
All right, next up we have our lightning talk. I'd like to welcome Jun Fan, who will be talking about DNA library prep from ultra low and highly degraded genomic DNA.
Hi. Oh my God. My name is Jun Fan. I currently work in the Texas A&M Institute for Genome Science and Society. However, this work finished when I was working in the Marshall Genomics Call. And this study was initiated between the collaboration between Marshall Forensic Science and the company called the Parabon. They initiated this kind of project because they want to bring the NGS technology into the forensic science. So we know the sample from the genomic sample from isolate from forensics, the bone marrow, like bone material, usually they are highly degraded. Also, the yield were very low. So to do this, this project just very initially for the trial test. So they start from we get the DNA isolated from the artificially aged bone material. Basically, that's the normal bone, like bone in the soil, and the UV treated. So just mimic the forensic samples. You can see that's the tegmentation, the trace. After we can see it only have the ring volume is very low, is around 2, even like that. But the genomic DNA still spread from the 1 base pair to the 3,000, like that. For increase the DNA library preparation efficiency, we fragmentate these samples, use the covariance, make all fragment surround as the 300 base pair. Because this sample is the actual degradation and also very low. Usually, the common, the DNA library preparation kit, they working for not work for yet. So the standard the DNA preparation kit use the adapter to the double-strand DNA, also have the until polished, right? Usually put the extra A in the DNA fragment. So we find we use the single-strand the library preparation kit that come from the one company they call the ClearBio. But I have to say, this job not sponsored by this company. So the product basically, they have the single-strand DNA. They make it from the library make from the single-strand DNA. They have the protein they call the single-strand binding protein, make no matter the double-strand DNA or single-strand DNA, they can make to the single-strand DNA. They also have the adapter they call the split adapter, make the liberation generation efficiency. So here we show the bioanalysis trace of the resulting library. So you can see I use the 3 different input, the 1 nanogram, 500 picogram, and even down to the 50 nanogram. So based on the bioanalysis the trace, you see they almost similar. So I mentioned that. That's from we initially work for the forensic science. So the forensic sample usually come a lot of the highly contaminate, like the bacteria, virus. So we thought like need the enrichment. So we, like the resulting library, we also use like, oh my God, sorry. Is like this one? Like, sorry, like, I'm sorry. Like suddenly, I just want like this one. Yeah, this one. Okay, that's the design library we use. We choose like the embryo, like the probe, the hybridization probe. So they do the hybridization. This one is like the final library. They come from the 1 nanogram, 500, and the 50 nanogram. So this also the bioanalytical trace. So you see even 50 nanogram, this shows a similar result like that. This like just like this library, then we put it onto the NextSeq 2000 in the 2x150 format to sequencing. You can see that's have the higher Q30 numbers above the 50%, 90%. This one is like the just show like the base across all the 300 the cycles. You can see ACGB evenly across like the 300 cycles. So far, like the data analysis is still in the going. So that's all I have the data because that's a lightning call. Maybe I'm too fast. So also I should analyze, acknowledge that's Dr. Primorno or Dr. Salisbury. That's my previous, like the director. I thank them for the support for this project. This also, the genomic DNA coming from like the forensic science, the Laura and the Tala. That's a group from the Parabons. That's my talk.
Thank you for that lightning talk. All right, let's just switch here. All right, I'd first like to introduce Bruce Kingham from the University of Delaware. After that, we'll be followed by Emily Quartemont, and both of them will be talking about sample prep and challenging species and quality. And then Shawn Polson will wrap up the session talking about bioinformatic challenges. Without further ado, here's Bruce.
Where do we just go forward? Okay. Yeah, first I want to thank Kelly for doing such a great job organizing. She coordinated and communicated excellent with us. And I need you close by because I'm not a PC person. So I'm going to tell you today about our experiences with DNA extractions. First question I have, who uses here PacBio and ONT? Thank you. So primarily what we're going to be discussing today applies to single molecule long read sequencing, not so much, maybe minimally synthetic long reads. Also optical mapping it would apply to. I want to acknowledge my team at University of Delaware because they actually do all of the work and help me develop this information. They're a very skilled set of scientists. I've been in genomics for 32 years now. First 10, I was 100% in the laboratory. Since then, I've gradually moved out to where 90% administrative duties now. So I'm mildly jealous and envious of them, but also at the same time very thankful for their skills. They have exceptional scientific skills, and they've also developed some specific skills that have generated an international reputation for us, primarily with extremophile samples as well as long read sequencing. And also along with Shawn Polson and his team, we've really built a very powerful genomics program at University of Delaware. So we are a basic research institution, and genomics research happens within multiple departments, dozens of departments, a wide number of colleges as well. We work with a wide array of sample types and organisms. We received our first PacBio in 2010, our MinION 2014. Probably 98% of our long-read work is with PacBio. And like most PacBio and ONT users, we are somewhat obsessed with high-quality, high molecular weight DNA. With the advent of hi-fi sequencing with PacBio, the high molecular weight is not as important as it used to be, but I would also say that if we have high molecular weight, it makes the whole process downstream considerably easier. So, second question I have, anybody tell me who this scientist is? Kary Mullis, exactly right. So if I were using today's political vernacular, I would say he is the enemy of long-read sequencing people. He developed the PCR reaction, and certainly it's undisputed that PCR has been critically important to life sciences. But the sacrifice, minimal sacrifice, I would say, when that first appeared, has been kind of the loss of laboratory finesse and craft with regards to DNA extractions. When I first started using PCR, it was common that I would just take cells, even a tiny minced piece of tissue, and put it into a tube with primers and PCR reactions, and I would generally be able to amplify whatever I was trying to amplify. High quality, high molecular weight DNA preparations and procedures are kind of a marriage between science and art, and the creativity that you have in the lab really yields results. A lot of the work that I did in the 90s, and some of my staff as well, we did a lot of work with bacterial artificial chromosomes. We did viral transfections. So we came with a lot of that experience with high molecular weight DNA. So this, DNA extractions can be very simple, they can be very complex, but basically what we're doing is we're taking a biological sample, adding some chemicals, maybe a little bit of heat, shaking it, hoping that the cells lyse appropriately, add some salt, add some alcohol, maybe spin it, wash it, and then what we hope for long-read sequencing is to get something similar to this. That is not as easy an effort as many of you that do this are aware of. A couple of easy steps to take, that we take, is we try to avoid standard pipette tips. We will use wide bore or even just snip off a very small part of the end of a standard pipette tip. If using a standard pipette tip with high molecular weight DNA, particularly if you're using force and delivering it quickly, you can actually shear high molecular weight DNA down to 25,000 base pairs, which for a lot of these procedures is not desirable. Secondly would be vortexing. Vortexer is deadly to high molecular weight. Anytime we need to mix solutions, it's just gentle inversions or a rotator. And thirdly is you got to try to pick the right people to work in the lab to do it. Sometimes that can be a challenge in an academic environment. Fortunately, myself and most of my colleagues don't have that same problem. So after we have this, the DNA, our goal is to obtain a solution that contains long fragments of intact double-stranded DNA and a low level of other cellular components. So cells are very complex with lots of organic and inorganic compounds. There are DNA binders and modifiers that are generally proteins or organic compounds that attach to DNA, can impede the downstream enzymatic activities. And the damage goes far beyond just strand nicks. There are numerous types of DNA damage that we can't see, we can't detect, and we may never detect that during the course of sample preparation, and it may not be noticeable or potentially theorized until we load these samples on the sequencer. All right, Kelly. Now I did something here. Nope, I got this screen up here. How do I? Okay, perfect. So I'm going to go through different sample types that we're regularly processing. I'm going to start with what is typically the easiest and move on to what's typically the most difficult. First would be animal tissue. The challenge is what we need to remove from these primarily. If it's cell culture, media compounds and residuals. If it is some type of body fluid, serum components, metabolites. And then some of the challenges, most challenging tissue types are lipids and connective tissue. Proteins and other DNA binding proteins are always going to kind of travel through that extraction process. The real skill is in removing as much of those as possible. Some notable exceptions with animal tissue are marine animals and basically anything slimy. Are incredibly difficult to sequence using long-read systems. And that's primarily because what carries through are commonly a large number of polysaccharides and those polysaccharides commonly will mimic DNA. And in the case of PacBio sequencing, the challenge there is that because those DNA or those polysaccharides can mimic DNA at some level, the polymerase, the theory, one of the driving theories is that the polymerases, which are mutated, can considerably bind some of those polysaccharides. And when we go to load these instruments, we get low loading levels and potentially short read lengths. Mitochondrial DNA can be a problem if you don't want a massive coverage of that organelle. So if it's possible to choose a tissue that has low metabolic activity, you can significantly lower the amount of mitochondrial DNA that you end up sequencing. The very best sample that I don't think we've ever had a problem in 12 years, 14 years with sequencing cell culture. Always get very, very high quality DNA, very high quality data as well. Second to that would be blood. You know, our recommendation is if it has blood, bleed it. we generally get very high quality data from that too. The next organism class, prokaryotes. Again, and this here, what I'm talking about are pure cultures. So it's important with pure culture prokaryotes to remove growth medium compounds and residuals. The challenge with these that you need to remove that are cellular components, polysaccharides, again, because they can mimic DNA, lipopolysaccharides, that has the dual blow of lipids, which are inhibitory, and then also polysaccharides, and then endotoxins, exotoxins that can often be protein inhibitors. Our recommendation is that you want to harvest at an early growth stage, and you want to grow enough volume to compensate for lower cell density, and wash the cells after they've been pelleted. Third would be plants. The challenges with these organisms are pigments, polysaccharides, polyphenols, metabolites. DNA extraction from plants introduces multiple challenges because of the very high levels of polysaccharides, phenolic compounds, and a variety of primary and secondary metabolites in the starting material. The term metabolite is usually restricted to small molecules. A primary metabolite directly involved in growth, development, reproduction. A secondary metabolite is not directly involved, but usually has an important ecological function. Plants have developed something akin to chemical warfare because they are not mobile. They need to protect themselves. So a lot of those compounds downstream can create quite a few problems with it. Our recommendations, you treat the plants for as long as possible before processing. That reduces the amount of chlorophyll DNA. It also kind of softens up the tissue, makes it easier to disrupt. Use young plant tissue, plant shoots. Young shoots are the best source. We disrupt tissue under liquid nitrogen. We have multiple mortar and pestles, and it's common we're in there grinding under liquid nitrogen plant leaves. And CTAB is very important with these organisms. Fungus would be next. If it's yeast, growth media compounds and residuals. Fungus have a high level of pigments, lots of metabolites, chitin, glucans, and then also proteins. So with fungus, you want to wash. If possible, I got to speed through this. Chitin is the second most abundant polysaccharide in nature behind cellulose, so there is a considerable amount of that in fungus. Insects, pigments, chitin, lipids, and proteins. Pigments, they're used in textiles because they bind things very, very tightly. They do that to DNA as well. We try to avoid the chitinous shell, disrupt under liquid nitrogen, high SDS and proteinase K, and we'll also use beta-mercaptan at times. Algae and environmental and extreme aphile samples, these are the most difficult. They have every challenge you could imagine. We provide no guarantees that it's going to work. After we have the DNA, we have to run through the analysis of it. We look at UV absorption for purity, fluorometric measurement for concentration, and then fragment sizing there for the integrity. I highlighted qubit and femtopulse because those are the most important for us. Femtopulse, from my perspective and most, is absolutely critical for long-read sequencing. What we don't like to see, this is a progression of a single sample going through library prep. The first, this is the original sample. This is RNA here. The investigator asked, it was a precious sample, asked us to go right through it, regardless of the outcome. So we did an RNase treatment. This is what we got before fragmentation. The size lowered significantly. That is a suggestion that there is DNA damage that will continue on as you continue processing the DNA. After fragmentation and before library prep, the size reduced even more, which is somewhat expected, but we had a lot of small fragments of DNA, which is problematic. And then after library prep, it was considerably smaller. So everything that we see that we don't like, we kind of consider it to be like a glitch in the matrix. That if we see something, there's and as we see more, there is the potential and the likelihood that there's going to be additional problems downstream. What we like to see is this, and I'm not saying we need to see it, but a very high molecular weight, tightly focused band. After fragmentation, a broader array, but there's little small fragment-sized DNA. After size selection, what we have is a fragment with a peak at 15 to 25 kb in length optimally. And with this was a Brazilian highly venomous snake that we had worked on a genome assembly project with, and we had outstanding results from that. So in conclusion, review the literature, cater your extraction procedure to the sample type, request as much sample as possible. The more we have, the more opportunities we have for success. Be as gentle as possible. Blah, 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 blah. I already went through a lot of that stuff. Don't resuspend in water. If it works poorly, try it again. DNA extraction is almost always going to be the most inexpensive part of long-read sequencing. So with that, thank you. And I'm sure I went a little over.
Thank you, Bruce. Next, we have Emily Quartemont from the University of Wisconsin-Madison.
Okay. Well, thanks, Kelly. And I'm Emily Quartemont. I work with the NGS sequencing core at the University of Wisconsin-Madison's Biotechnology Center. And in keeping with the theme of Bruce's presentation, here to talk about the key to long-read success being in high-quality, high molecular weight DNA. So before jumping into anything, just an overview of the long-read specifics that our facility supports. Got a list of the long-read services that we offer, and in terms of sequencers, we have 2 PacBio Sequel 2s and Oxford Nanopore's Promethean AP24. So success in your long-read workflow is going to start with your successful isolation of high molecular weight DNA. Our facility has 2 main kit types that we use for the majority of projects that come through our facility. We use what's now PacBio's nanobind disks for those sample types listed, and also QIAGEN's genomics tips for the other sample types listed up there. The off-the-shelf protocols for both of these kits have been really great starting points for us, but I will say that as we've gained experience with varying sample types and sample conditions, we've made some kind of standard modifications to our versions of these protocols that have helped us improve yield and quality. One to point out with whole blood, we pretty much for just about every project that comes through, we'll use an upfront RBC lysis, and we've seen that that greatly improves quality. But I say standard modifications with a slight asterisk that, you know, it's not going to work for every project that comes through. We've dealt with samples that come to us that have maybe gone through a few more freeze-thaw cycles than would be optimal, and so we have to think maybe there's been some premature lysis, and so trying to pellet your cells and take that through is not going to be as successful. And we also often see blood come to us from mouse samples with the animal core that's in our facility, and oftentimes they want to keep the mice alive, and so volume is a limiting factor there, and so we'd rather take the smaller volume just in as whole blood rather than doing an RBC lysis on a smaller volume of blood. And we know that quality is going to be a little not as great, but at least we know why. And in terms of tissue, we use tissue washing protocols for any samples that are going to come to us that are buffer preserved. The nanobind kits have a really great application note for samples that come in various buffers. As any bit of that residual buffer that's going to travel with you through extraction is going to have a little bit of a negative impact on the quality of DNA that you're going to get out. So with that, generally speaking, fresh snap frozen tissue just always does better. And so if you have the option to kind of influence the type of tissue or the preservation method that any of your customers use to send you tissues, fresh frozen tissue just does better. But then that also brings up the question of, what are the best tissue types, or maybe what are the tissue types to try to avoid. And at our facility, we try to avoid eye tissue often because lots of problems there. If you've got organisms where you can, with the scope of the project, try to avoid the digestive system, that's usually a good thing to avoid. Sometimes tail and ear tissue can be tough to work with, but sometimes customers would rather use that because then they can keep the animal alive and send that to you. And I will say that through this job, I learned way more about the anatomy of a spider than I ever thought I would. But I have learned that the legs and thorax are best off of spiders and other arachnids and things like that. So always something to consider of what's the best tissue type that you can get from a customer. When it comes to the more typically problematic sample types, a couple of those up there with an asterisk. We try to suggest pilot projects wherever we can. It's usually better for us and the customer if we can start with a small subset of samples, particularly if it's a sample type we don't have as much experience with, to help us identify where some of the extraction pain points might be so that we can successfully isolate DNA before jumping into a full-scale project with lots and lots of samples. Then, though, the question is, where do you even start to troubleshoot? Lysis is often one of the most difficult hurdles to overcome, and we've got kind of three basic adjustments that we will make: time increase to your lysis incubation, temperature increase, and buffer adjustments. Particularly for the QIAGEN genomic tip protocols that we use, we make our own lysis buffer in-house, and they're slightly different based on the sample type that it's going for, but they're all CTAB based. And so considering what types of things you might be able to add to the buffer or take away for a different type of species is always something to think about with this particular sample at hand. I would say kind of further down that lysis list is that if you really can't crack it open, liquid nitrogen grinding will do the trick. And then, grind it to a powder and take that into your normal protocol. And also that's up there is enzymatic pretreatments. And an example within our facility, we don't necessarily have a fully vetted gram-positive extraction protocol, but we do know that they're more difficult than just our gram-negative bacteria to extract DNA from, so using our normal protocol doesn't work, and so we add often a metapolyzyme pretreatment to gram-positive samples to help them successfully work through the our gram-negative protocol. So once we have DNA extracted, we use a 3-step QC process utilizing the Thermo Fisher's NanoDrop 1 qubit, which we run on Promegis Quantus, and then the Agilent femtopulse for sizing. And what I will stress here is that all three of these things are really needed for us in combination to get as full of a picture as we possibly can of what the DNA actually looks like before we try to go into prep. And the little Excel screenshot that's up there just gives you a view into our really basic internal flagging system that we use just to bring our attention to some values that may or may not be of concern to address before we start prep. So particularly, where nanodrop ratios just highlighting if they're below what is considered pure, or highlighting a concentration discrepancy between nanodrop and qubit. So once we do QC, we've got kind of 4 major red flags that we look for, and I've mapped our kind of basic thought process on how we might address those red flags, either in isolation or in connection with one another. So if we're looking at just a low 260, 230 by itself, generally we're just going to press on. It could be something as small as a little bit of guanidine carryover from extraction that could affect this number, and that's not something that's going to really have too much of an impact on your success of library generation for long reads. Next thing that we're going to look for is a concentration discrepancy with a low 260, 230. If we see these two things together, we've got extra questions to ask, largely around what inhibitors might be there and how are we going to go about combating those before we proceed into library prep. One of the first questions that we ask if DNA is customer extracted and submitted to us just for prep is were these samples RNase treated, because that's one thing that's going to spike your nanodrop concentration and affect your 260-230 ratio. That's not something we necessarily have to go back to extraction to fix. We can use something like an SRE. It's a short read eliminator clean. It's a PacBio product, which I will circle back to in a little bit here. There is also always the option when we see these two things to press on, which doesn't come without having at least an informed conversation with your customer to go, look, we see these risks, but we get it if you need to press on, and that's great, but there may be some effects on your library preparation and the sequencing success. The third red flag listed up there is the low molecular weight DNA, or what may appear to be low molecular weight DNA. And this we can either see on its own or very often in combination with a concentration discrepancy and some funky nanodrop ratios. A couple ways to combat this: your good old-fashioned bead clean, or we also often use this SRE clean. And the last thing that's listed up there is heterogeneity issues. There are a couple places that you're going to see that this might be an issue. In your femtopulse results, we can sometimes see a really nice peak that is your sample, and then there's some even larger noise up above the 200 kb range generally. That might be a hint that there might be some heterogeneity issues. But then there's also just looking at the physical sample, and is it super gloopy and snotty, which just on a basic level can have some issues of pipetting accuracy that you want to make sure you address before you try to go into prep. So, we often use heat to help with that. We have kind of two methods that use kind of different temperatures, 37 heat to help relax, or 55, both seem to work. But otherwise, also to help combat this issue is to introduce some shearing. That's baked into the PacBio library prep, so you're good to go to just proceed that way. But for ONT, for some projects, we can add shearing up front, either needle shearing or with the Covaris G tubes. And then I also threw up there's the extra little consideration for which ONT kit you might be able to use. We use the LSK114 and the RBK kit, and often the rapid barcoding kit can be a little more forgiving, and so if that opportunity is there for the needs of the customer's project to use that kit, we might suggest that, just as it is a little more forgiving. So circling back to this SRE, short read eliminator, there are kind of three versions of it based on the size that you're going for, and we often only ever use the XL for ONT purposes. But the example that I've got up here, these are some metrics that were aimed at PacBio. And if you look at the first three samples, that's pre-SRE, all of our nanodrop ratios super low. Got a couple of crazy concentration discrepancy issues, particularly with C. And then if you look at the post values, we had a significant improvement in our nanodrop ratios, and we've eliminated, at least for one of the samples, that concentration discrepancy. And although these nanodrop ratios are still in the red, there's a significant enough of improvement that we will feel better about proceeding into a library prep with these samples. But if you look at sample B, no improvement. Arguably, it got worse post-SRE. So then the question of, what's going on, and that's where the femto comes in handy. So if we look at the sample A that worked just fine, we had some low molecular weight noise. The SRE successfully removed all of that, and the post-SRE looks great and fine to move into a PacBio library prep. But B looks almost identical, which should not be the case once you perform an SRE. It does work. So instead, this tells us that that's likely not actually low molecular weight DNA, but more than likely, it's higher molecular weight DNA with a charged molecule or inhibitor of some sort bound to it that's affecting the way that it runs on the femtopulse. And that's something that we have to circle back to extraction in order to address, but should address before we would try to move into a library prep with this particular sample. So, and with that, I will just say thank you and pass it off to Shawn.
Thank you, Emily. Next, we have Shawn Polson from the University of Delaware.
All right. So thanks, Kelly, for the invitation to talk, and thanks for all of you for coming out. So before I talk about challenges with long read, I wanted to at least say something about a win. And in addition to running a core at the University of Delaware, I also run a research lab in collaboration with 2 other faculty. And we look at bacteriophage and other microbial viruses in the environment. And PacBio in particular, but long read, has been a godsend for the type of research we do. We're using metagenomics, we're going out looking at environments for viruses, and you think they're small, Illumina should be enough, you can assemble them out, but in reality, you go out in the environment and look at viruses, and there are a million variants of every possible thing there, which for a short read assembler is like kryptonite. So having long reads where we can actually see which genes are occurring together in certain viruses has really changed the way we can do research. We've definitely had some input issues with, having to do some massive concentration, but in general, long reads have been great for us. We tend to go out, look in the ocean, look at different environments and see what these viruses are doing, and it's really helped us to make what we like to call, some genome to phenome types of conclusions from our data, because observing viruses doesn't give you much. You've really got to have a hook to talk about them, and the genotype really gives us that. But okay, so on to more of the point of the presentation. I run a bioinformatics core facility at the University of Delaware, with a staff of 6, and we do all the common things, looking at different types of bioinformatics, lots of omics. And as Bruce has already alluded to, we are not the common mouse human type of facilities. We handle those, but we probably do that. A model organism for us is an unusual project. We do a lot of non-model organism, and they come from all over the place. So some of the challenges we see may be because we're kind of on the edge with some of the organisms we're looking at. So in general, challenges with long read for bioinformatics, we can begin with the elephant, which is always looming in the room when you talk about long read. And I think most people probably have a good idea of this by now. It's always been the accuracy. And with both PacBio and Oxford Nanopore, this accuracy issue has really started to go out the door. Is it completely, out of scope? No, there's still some issues, but it's not the issue anymore, right? So we can move on to more interesting things rather than having to worry about how to tune the technology and the analysis to those types of things. So when I think about problems with long read analysis, it's less, what's going wrong with the technology, and it's more what I call the mess of success. What is the technology letting us do now that, before wasn't possible to do at scale, and why are we encountering things now that technology is essentially putting in front of us? And importantly, we're bioinformatics experts looking at this data. We notice when things look a little weird and we dig further down the rabbit hole. But we have plenty of investigators who like to do the analysis on their own, and how do we help to guide them so they know what to look for to make sure they're not running into some of these types of issues. So general challenges we see, there's pretty quickly evolving technology and software. It's calmed down a little bit, but you can still look in journals every month and see new tools for doing different types of analysis and long reads. It's really hard to know what software to pick, what to use. In practice, when we do something like genome assembly, we do it with multiple different tools because we found certain tools work better for certain genomes than others. So it's not like a one-size-fits-all type of solution. And there's still a lot of that with the bioinformatics software kind of catching up with the technology and trying to figure out the right way to apply it. I'd say for us, and maybe this isn't normal, but because of the unusual things we do, the standard projects are more rare. And for us, it's an ingredient for the kind of rabbit hole projects that are sometimes fun, sometimes frustrating, but don't always work well in a fee-for-service model when you're trying to give some idea of how much it's going to cost to do an analysis. So that's a definite challenge for bioinformatics with long read and short read, but I'd say at this point, short read has become much more predictable. We run into these problems more with the long read. So I could talk about this from different perspectives, but one that I thought of that we've kind of seen a lot, and we're seeing increasing people asking for it, is microbial community analysis using long read. So I thought I'd talk through that a little bit. So you probably have some basic idea with microbial community analysis. A lot of times 16S is kind of used synonymous to amplicon analysis. It's the ribosomal RNA, the small subunit, and it's great for doing microbial ecology because it's got some really conserved regions we can make primers to, and it's got these variable regions, the different V regions at the bottom that, they're variable, which means we can see differences between different types of microbes. With Illumina, the 1,500 base pairs of the gene were not really in play. We couldn't look at it all at once with short read. So most people were looking at a region called V3, V4, which gave, fair resolution. But it was really more like genus, and it varied depending on the taxa, how good that resolved things. And with microbes, the difference between strains can make all the difference. The difference between species, it could be different sides of the planet diversity-wise. So there's a lot of issues with trying to get to the level of resolution that give people the answers they're looking for. And then with these technologies, you don't have to stop at just looking at the 16S gene. Long read lets us look at all 1,500 base pairs, but we can potentially go beyond. And one of those tales I want to tell today is kind of related to that. So to give you a quick background on, what we're doing with microbial community analysis, usually, almost always, the point is you want to compare one set of samples to another, right? And to do that, you've got to have buckets of similarity. You've got to say, in sample A, you've got 10 of thing, whatever it is, E. coli. In sample B, you've got 20 of it. So there's some difference there. Turns out taxonomy is not the best bucket to put things into. It's imprecise. It changes. So we usually try to use something that's more sequence-based, like traditionally operational taxonomic units, OTUs. It would just be anything that's 97% similar, put it in a bucket together. More recently, with short read, especially a different flavor called amplicon sequence variants, where instead of trying to cluster things together at 97%, we're essentially trying to do it at the sequence level, 100%. No variance. We just take all the different repeat reads we're getting back and we count those up. And usually there's some aspect to trying to correct sequence errors, which is a little bit of bioinformatics black box, but it tends to work. So just to kind of show this visually, you see over here on the left, real sequences. That's kind of what the sample had. In the middle, you kind of see what you get out of sequencing, which is those original, maybe this will work. The originals are here, but you've also got this cloud around it, which are mostly sequencing errors. So you take the OTU and you get rid of the sequencing errors, but you also end up combining some things together. Not always a good outcome. So if you want to look at it another way, if you have this colorful cast of characters, you can OTU cluster them into things that kind of look the same. But you might notice that You know, the mice and rats have merged here. The representatives maybe not the best representative for the group. And so there's a little bit of difference going on here. With ASVs, we try to get around this problem by correcting these errored reads and putting them back into the appropriate group. And hopefully we get something out that's a little more reflective of reality. Cartoon analogy, you get a better clustering, and maybe that error correction actually gets you back to something that looks like the organism that's really there. So what's the problem? Why don't we just use ASV with PacBio and go forward? You know, it turns out the error correction software, it's sequencing technology tuned, right? So we knew this from the beginning. There's got to be some tuning for using PacBio per se with it. And some of the tools, .2's one, they've made models for doing PacBio CCS sequencing or hi-fi sequencing. So it's there, the ability to do that. But we see some weird things going on sometime. I'm going to say too high resolution. I'll kind of get back to it. But to get back to a thought that I planted earlier, you know, do we only sequence the 16S or do we try to look beyond that? If we look at the 1,500 base pairs of the 16S, that gives us a good idea of diversity, but it doesn't always give us a species identification. It's still not always good enough to do that. So there's a lot of interest in looking at this ITS region downstream, which, you know, you can make primers that flank both the 16S and part of the large subunit, get amplicons for that. And that includes this ITS region that, for all intents and purposes, has no evolutionary pressure, which means lots of mutations can accumulate there, and it's really easy to differentiate things. So there's some problems with that. Taxonomic databases have kind of been built for 16S. There are ones out for longer regions, but a little harder to use. The molecular clock varies a little bit. When mutation rates different in different parts of what you're looking at, that can impact those ecological measures that people are trying to get out of it. And also, it can kind of confound the error correction software because it's expecting a certain rate of changes, and it can start to think that real diversity is actually, you know, sequencing error and try to correct it. So this issue I keep raising of resolution being too high, what do I mean? Microbes with different ITSs can be almost identical phenotypically. The ITS can actually mutate so freely that, you know, functionally identical things will look different. So that's a problem because that means when we're looking across samples from different locations, there's going to be nothing shared in the bucket because there's little differences in that ITS region, which means we have nothing to really compare across samples. It's really obvious when we have a longitudinal sample where we're sampling from the same organism over and over. We'll see easy ASV connections between those individuals from the same sample, or the same samples from the same individual. But when we try to look across individuals, there's a very few connections going on, which means our analysis is very difficult to do, but there's usually a few. And that means if you don't know what you're doing, you can actually move forward with the next steps, make visualizations based on just a few connections between those samples, which can give a very erroneous view of the microbial community. So that's one kind of snippet from microbial community analysis. I talked a little bit about doing that longer amplicon. People have started to do that. They are still useful. If you're trying to source track a microbe, that is the way to do it. You know, that's a great way to do it. If you're really interested in looking within an organism and sampling it over time, it's a great way to do it. But if you're trying to look at a broad microbial community from different locations, different individuals, there can definitely be some challenges there. And at times, even if you're just looking at the 1,500 base pairs of the 16S, some of those connections still get lost. You still get some resolution issues. So even if you're not trying to do that longer amplicon, you still can run into some of these things. All right, so quickly, it's similar for other types of approaches. We have these conflicts of the traditional approach that was largely short read sequencing built and how we do it. But we also have things like genome assembly where, you know, long read has revolutionized genome assembly, right? It's so much easier to get a decent quality genome now than it was just a few years ago. Older, like PacBio, pre-hi-fi You know, we definitely had issues with the indels from the reads sneaking through to the assemblies. That caused frame shifts, genes got disrupted. Hi-Fi has largely fixed that, but we still see some weirdnesses. So I'm getting short on time. I'm going to skip this slide so I don't have to explain it, but I'll go over it briefly. What we tend to see is, you know, we're looking at a lot of weird organisms, and a lot of times they have some weird biology going on. And what I was just going to show, it was an Aedes mosquito, but it was, you know, Aedes carry yellow fever, Zika, dengue, big disease vectors. And most people know about Aedes aegypti. This was a very divergent Aedes that we were looking at. We didn't know that at the time, but we started sequencing it, and we started to find it really odd. Like, we could see all the different genes in the genome, but no matter how much we fine-tuned our transcript calling algorithms, we couldn't actually get intact open reading frames for a large number of the genes that we expected to be there. And for others, we saw way too many copies of them. And it turns out with this one, the weird biology was that well over 50% of this genome was retrotransposons and other mobile elements. So there was a lot of active shuffling going on in that genome. So that just led to a really difficult to assemble genome. You know, we were using PacBio Hi-Fi. With Hi-Fi, you're going maybe 20 kb fragments, and in some cases, that's just not enough to resolve those types of issues. So we see that happening. We've also seen it in cell lines that are used in manufacturing, the ones that, you know, not the ones you're using in common human mouse, but rather ones that have been force evolved over time, kept in culture for 20 years. Weird things going on with their genomes. You try to do these assemblies, you try to do the annotations, and what you get back, it's really a crapshoot. It's very difficult to deal with, and we see lots of issues with them. So, you know, occasionally you still get some indels passed through from the sequencing, but by and large, what we're seeing is weird biology leads to weird things in the analysis that we do and the assemblies that we do. So, some take-home points, you know, long reads are letting us do stuff we couldn't before. But we're exposing a lot of edge cases where we see weird biology, and that leads to a lot more bioinformatics effort for the people that are trying to analyze and say something about it. Software is still catching up, but really that biology is key, and I think this is a good take-home for the bioinformaticists is, you know, a lot of these things we track them down from repeated conversations with the investigator because they know their biology and they can help us to kind of figure out why is it weird, what's going on, is it sequencing issue, is it assembly issue, or is it something else? All right, so thanks to everyone who helped, especially Maddie, who is the long-suffering genome annotator in our group, and I guess.
Thank you so much, Shawn. So we have a few minutes left in this session, so I want to open it up for some discussion and some questions, if anybody wants to raise their hand.
Oh. Analyze the 16S and ITS together? Is there a specific pipeline they use or developed, or is there a standard procedure with it?
Yeah, I think it's still evolving. What we tended to do, we would take one of two approaches, and we kind of experimented a little bit with them, is we'd essentially either make the ASVs just for the 16S portion, but we would use the full portion to do the taxonomic identification, or we would make the ASVs for the full length, but then we'd use like a 99.5% OTU to kind of go on top of that and connect similar ASVs to each other so that we could say something about the ecology. And both have their pluses and minuses.
So is there a way to separate that and build, for example, a phylogeny only based on 16S or ITS separately, or do you analyze it always together?
We do it both ways. So we do, like, we will artificially cut it at a certain point to talk about that. And it depends a little bit on what we're studying at the time, what makes most sense.
Oh, hi. Noi from MIT. Just a question for Bruce. So you said do not resuspend in water. I agree with that. There's a bunch of buffers we use, but is there like a universal buffer that you think is best for suspension, or does it vary based on organism, tissue type, cell type?
No, it doesn't vary from my perspective. We haven't looked at that closely enough, but tris, 10 millimolar tris, and in our hands, particularly with high molecular weight, the mistake most people make is not letting it resuspend long enough. We'll oftentimes let it sit on the benchtop for days.
Hi, I have one question for Bruce and one question for Shawn. Sorry, Emily. So I'm a mathematician, so I learned a lot of things today. So my question for Bruce is, you mentioned marine animals are hard to deal with. I kind of understand why slimy things are hard, but marine, if they're vertebrate, right? So why marine animals?
The compounds that they carry over, glycolipids, polysaccharides primarily, and then also you have to consider that a lot of these marine animals, and a lot of animals, they develop mechanisms to protect their DNA based on the environment that they live in. And primarily the way they're protecting their DNA is by physical mechanisms, a lot of binding proteins. And so when we extract from those tissue samples, it can be exceedingly difficult to remove those binding compounds from the DNA without also damaging the DNA. Commonly, if you're trying to remove something that's bound to DNA, the stronger it's bound, the more affinity that it has for DNA, the rougher you have to be on it to remove it, and so hence you're going to damage DNA after that.
Okay. My question from Shawn is, you mentioned for the 80s, have more than 50% of regional transposon, and that makes problem for the gene annotation. But I think human also have like 50% of transposon element. But why is that 50% of regional transposon in 80s making it so difficult?
Yeah, so I think in the 80s there was more active mobile elements going on. It actually had several, I skipped talking about it, but there were several large duplications in the genome as well. So we saw about 20% or 15% of the should be single copy genes occurring in duplicate, and looking through it, it looks like they actually were duplicated, but sometimes the reads just weren't long enough to put them in a duplicated position, so sometimes you would end up with duplicated genes where you'd end up with the reads stacking on top of each other, and you get variants within the gene that sometimes can cause a stop codon or a missplice site.
So my question is kind of a general question. Honestly, it's directed to anybody in this room who has insight. But is anyone using something other than gel plug lysis protocols to get ultra-long, high molecular weight DNA from plant samples? All right. Gel plug lysis it is.
There's
Awesome.
your
Thank
answer,
you.
Sharon.
Thank you for the talks. They were very good. And Mark Van Eden from Zymo Research Corporation. We specialize in trying to get DNA out of things. You're talking about these analyzers that are very, very precise. We have them as well for long read, short read, etc. The ramifications of how samples are collected and stabilized and extracted are profound. Additionally, then you start saying, how are we getting the DNA out with art? Not everybody can perform art. What is the standard? What are the standards or references that are being utilized so we can tell if labs are artistic or not prior to their sequencing?
I don't know if there are standards, considering that there's no standard to species, and there are so many different characteristics among species and within species based on what environment that they're in. So yeah, that's a difficult question to answer. You know, I would add, you said you're from Zymo? Yeah, and so when we're extracting DNA, most of what we used to do was homebrew stuff. But credit to Zymo, Omega is another, QIAGEN has also done it, where particularly in the long read era, they've really come up with some very effective means for extracting nucleic acids from various different sample types. And we'll commonly kind of take your kits and the other ones and just tweak them a little bit. But yeah, they've become very effective and they're getting better.
My question is for Emily. You mentioned that you improved lysis using some enzymatic pretreatments. Are there any in particular that you found especially helpful?
The one in particular that I can talk to is just for gram-positive bacteria utilizing metapolyzyme. We've done overnight heat treatments with metapolyzyme, let something sit for hours and hours and hours, and then take that in, and that seems to help, particularly in that case. That's the kind of big example I think that I can point to.
So I'm a flow cytometrist running a cell sorting facility, and been sorting nuclei for traditional sequencing applications for a while now, and we know that using DAPI and other fluorescent DNA labeling techniques upstream of those applications is not a problem, but I'm wondering if that would be a problem for long read DNA preparation, having to remove those kinds of molecules. If you have any.
I would say maybe.
Okay. And so then, so a pilot experiment would be warranted?
Yeah, and not necessarily would that even answer that question. You know, every project is kind of its own individual. But when we're processing, and Emily would probably agree, I mean, there are multiple points during that procedure where we are washing. And of all the compounds that we talk about, we've spoken about that interact with DNA. Some of them are removed fairly easily, some of them not.
And there are multiple fluorescent DNA intercalators that do intercalate differently, and so depending on the flow core that is preparing your upstream of what you are getting, there might be flexibility in what they can work with, which is also partly where my question is coming from.
This is a question for Bruce and Emily. You talked about polysaccharides or chitin being challenging. Have you tried doing any enzymatic treatments to remove? What are your strategies for trying to address contamination with polysaccharides or chitin?
I mean, we just kind of throwing everything that we can possibly think at it to try to get it to work. There's not really a set process that we necessarily go through. We'll try what we have kind of as our standard, and then if it doesn't work, we'll go, well, what else can we throw at it? What else is in the freezer that we've got that we can throw at it, and just kind of see what we can make happen. So there's I don't really have a great standard answer of what we do every time kind of thing.
It's nice that, I mean, Emily and I didn't talk about this beforehand, but we use kind of the same process. We try. If it doesn't look right, we try again. If we try something and something doesn't look quite right, like I said, we look at it, like a glitch in the matrix. If something doesn't look right, then that's kind of a red flag that, downstream, there could be problems with it. Chitin, I think, falls in that category of polysaccharides that at least with PacBio, we think may mimic DNA, and the polymerase binds to it, so then there's less polymerase that is available for binding DNA and sequencing.
Thanks. Kelly, do I have time to ask Shawn a question too, or do we need to wrap up?
We have a few minutes before lunch, so I'll let everybody, if you want to scatter out for lunch, but feel free to ask.
Shawn, how do you think about have you seen an increase in people wanting to assemble mags instead of doing a 16S or other study? And how do you help to guide that decision-making process?
Yeah, I mean, we definitely see people wanting to do that. A lot of times it's kind of, what's your goal? A lot of people for years have used 16S as a proxy for what type of activity those bacteria might have. And at this point, if that's really what they're interested in, is getting to that like functional activity, metabolic level, then they definitely need to be going the MAG route. And that's, I mean, I think that's the kind of steering point for me is, are they really just interested in who's there and, you know, what's differential, or are they looking at a functional component?
This question's for Emily and Bruce. So I was kind of curious about like your experiences with the nano drop and looking at those like 260, 230 ratios, because it's been kind of like debatable like how useful that is, right? Because we've seen sometimes it looks terrible, but the sample sequences very well, and sometimes it looks, you know, good and the sample will just fail. So are you guys finding it actually like a useful metric? Because honestly, we haven't seen too much correlation with those.
No, I mean, we do it because it's very simple.
Yeah.
And it gives us just kind of a very quick snapshot of what we have. It takes a minute to do it, but like you're suggesting here, it really doesn't mean a lot. We've had samples that have very low 260, 280, low 260, 230 that work just fine.
Yeah.
We've had samples with high 260, 280 and 260, 230 that work terrible. And that very well could be, even though it might be a very minimal level, if the failure is due to an organic compound or a protein, it may be a very strong inhibitor of enzymes.
Okay.
I think
Thanks,
just
Emma.
to add on top of
Yeah.
that, it's something, we don't necessarily use it in isolation. That's why I say it's important that we use all three of our QC processes together, because I think in combination, it kind of informs how we address issues that we might see with our other two QC metrics kind of thing, but not as useful necessarily by itself, but in combination with Qubit and Femto.
Yeah, that's great. Yeah, so you're not like ruling out a sample just alone on that. Yeah.
NanoDrop,
Great.
yeah.
Shawn from Zymo Research. I have a question to Shawn. I'm speaking on behalf of my microbiome team. So when you're talking about 16S sequencing, talking about data, a lot of error may come from sequencing error. So actually, our team also facing, like when they present the data to me, I was so shocked. Like it used to be Illumina sequencer, they are 300 pp. The R2 quality is not that good. So luckily we got chance to, our facility got chance to have their access to our new chemistry. I'm not Illumina person, but I just share news to everybody here. Their new chemistry, I look at R2, the Q30 getting pumping up very good. So that will be beneficial for downstream like analysis. So we just correct error at a wet lab stage. We don't have to bother in bioinformatics to correct, use hardcore bioinformatics too.
Yeah, absolutely. I think anytime the error correction can actually come off of, you know, the sequencer rather than having to be done bioinformatically, you're going to get a cleaner result. The error correction algorithms that do the ASV process I was talking about, you end up with a lot of sequences that go into their I don't know bucket and kind of get taken out of the analysis. It's not always clear unless you're really looking at it, but there's a fair percent that get filtered out during that process just because the error correction is too uncertain. So I think anything you can do for the upstream accuracy to improve that is going to give a more complete picture.
Yes, thank you. So my colleague will talk on behalf of Illumina. There's 6 p.m., so I won't be there, but anyone interested about microbiome study, so welcome to that section.
Okay, we'll take this one last question.
Hi, this is for Emily. You showed your A and B. So in the B situation, can you expand a little bit into what did you end up doing? Did you actually sequence it, and you know what's the solution for that?
Sure, we did not end up sequencing that particular sample. In this particular instance, it was a customer extracted sample, and so it's not something that we handled the troubleshooting of, and so it went back to them with as much recommendation as we could give to say, there's something here that needs to be remedied. We'd suggest looking at your protocol and seeing what adjustments you can make before sending stuff back to us.
Do you see that often? Because we've done a lot of them at the University of Minnesota here, and the first time around, actually, a couple of weeks ago, we came across that situation, so that's why.
This particular example was a first for me to see.
Okay.
So it's not as often that we see, but I also see we do get more projects that use us for extraction, and so, and we don't, our protocols, we don't see that issue as much with our protocols. It's more so we don't know what customers are using for their isolation to affect that, so.
Bruce, do you have anything to add for that?
I'm sorry, I blanked out. What?
Well, in Emily's slides, it showed that,
Yeah.
That there was, it got worse after the
Yeah, so
cleanup, or it's much smaller material, but it doesn't look like it's small material.
Yeah. Our standard in those cases is we communicate and interact with the investigator, and with him we make that decision. We try to make some kind of prediction about success versus failure. But that obviously can be very difficult to do. We deal with a lot of very precious samples, extremophiles taken from the bottom of the ocean or something else. And in many cases, these investigators have the funds for it. We have a sample that they may or may not get data for, so, why not try it? And commonly our predictions are pretty correct. One thing I've gotten really good at is laying out disclaimers for that.
Yes, yeah, that's right. Yeah, thank you.
Are you all
I'm
set
fine.
for one more quick question? Yeah.
You said last one, but
Yeah.
it just occurred to me. Sorry for Bruce. Do you ever notice if, or how can I ask this? So if it looks like your sample prepped really well, like your QC metrics are spot on, your femtopulse looks fantastic, you put it on the machine and you get a third of data that you expect, what metrics do you look on the machine to try to argue that that was like a sequencing reagent problem? And do you notice that happening a lot with PacBio?
It's gotten much better. We do see it sometimes. Those projects where everything looks good and the sequencing performance is poor are rare, but they do happen. And it depends on what the metrics we're looking at are. The primary metrics we're looking at is loading percentage. Those are the number of wells that have an actively sequencing polymerase. And then we're also looking at polymerase read length. So if we have very low loading, but we have long polymerase reads, that suggests that there's not DNA damage, but in that case, a lot of times what we are theorizing is this DNA mimicry that I've talked about. Every organism has polysaccharides in it. We think that those polysaccharides can mimic DNA, the polymerase binds to it, so, and they're not going to generate data. Those polymerases that do bind DNA are going to generate long read lengths. So that tells us that something is inhibiting the polymerase from capturing DNA. Whereas if it's a low polymerase, short polymerase read length, that suggests DNA damage.
Yeah, if I could just add to that as well, if the polymerase is not getting around a full 3 times, you'll notice that in the QC metrics in the run report, and that is typically indicative of some type of nick in the DNA that we weren't able to see prior to sequencing, and that'll really drastically impact the yields of your sequencing run.
Yeah, we have controls that we run, low spike in levels that typically we can effectively use to troubleshoot. If our control sample, the data from that looks the same as a poor performing sample, maybe low loading or short read length, that could suggest flow cell issue, maybe chemistry or fluidics or pipetting. I mean, it's a very complicated, advanced instrument. So, like all of the other sequencers, things can go wrong that don't have anything to do with the sample or the operator or the extraction or the library prep.
All right, and that brings us to lunchtime. I want to thank everybody for joining and participating. A big round of applause for our speakers. And as a reminder, I do believe today is the therapy dog day, so make sure you go and enjoy the pups.